Hi everyone, welcome back to discuss a part 3 for formulation development. In this, we will discuss certain part of what are the importance during the development with respect to the dosage forms and what are the tests to be done and how this method would be done for the testings and what are the stability conditions to be considered as a part of your developmental activities. First of all, we would like to just discuss what are the various test method is being used for pharmaceutical product, because test methods are very important for every stage of your developmental activities. HPLC, which is high performance liquid chromatography, is being used to quantify a drug substance acid, degradations, impurities. Gas chromatography also is being used to quantify residual solvents and certain level of headspace also shall be quantified using gas chromatography. Mass spectroscopy, that is being used to identify the mass identification, because basically this all is being used as an identification tool for many peptide components. In addition to that, the FDA guidance says from 2018 onwards, every drug substance and excipients should be characterized for elemental impurities, where the importance of inductive coupled plasma with an OES or MS, a mass spectroscopy, would be added advantage to develop a method and test the samples for the drug substance as well as the drug product as well. Apart from that, you will be having certain minor test parameter, which is pH, by using a pH meter, a color and clarity by using a UV or quantification technique of color by using a color band, or a turbidimetry to determine the clarity of the solutions, and as well as the osmometer. And there are certain cases where you need a headspace oxygen analyzer to define your headspace. And for tablets and capsules, you may have to do hardness, thickness, friability, disintegration time, dissolution. So these all various test parameters and equipments are needed when you test your final formulation. Here, a specific test required for the pharmaceutical dosage form. What are the specific tests? We have not generalized here, but we have put a specific test needed for each dosage form. As we discussed, for the tablets, you need a hardness, disintegration time, dissolution rates, and friability. Capsules, dissolution rates, pH, hard gelatin, inspection for leaks, soft gelatin, visual examination for brittleness, suspensions, viscosity, effects of freezing and pH, ointments, homogeneity, viscosity, effects of freezing. Oral powders, completeness of solutions or dispersions, and implants, hardness, dissolution rate, and friability. So these are the very, very specific tests for each dosage form, apart from the regular test parameters being considered while developing the product. And injections, sterility, syringeability, effects of freezing, multiple insertions for the multiple doses, isotonicity, a color and clarity, and pH as well. Of course, there are few solutions have to be done a same test. In addition to that, a microbial attributes, a sterility or BET, a viable. Aerosols is a peculiar test where we have to do a delivered dose, particle size distributions, wall corrosions, spray pattern, etc. There are few lifeless products where you have to perform a reconstitution time, a dilution study on storage, and reconstituted solutions on storage, or very important test parameters for the lifeless product. So these are test parameters has to be considered as a part of your developmental activities. In-process testing parameters for pellets, because pellets is being used for most of the products where you can do a spear. Using spear you can coat the pellets and create your drug product into the pellets form, and those pellets shall be filled into the capsules. For example, a cold act is one of the greatest example which you guys have knows very well. So these pellets, a description of pelleting conditions, a moisture content, a drug assay shall be reported at initial, intermediate and final stages, because it varies each stage as this pellet process grows. Sterile solutions. It is mandatory, it has to be done prior to filtration and post filtrations, because post filtration there are a lot of chances your drug content or preservative get alters. And then tablets. All proposed to premix and finished product feed products, top, middle and bottom should be sampled during in-process conditions, because the homogeneity is very important as we do a mixing of various components with a different particle size. So the blend uniformity shall be tested at different stages of manufacture. So normally here we just presented here how a sample test request format, which contains your test request, your sample ID, your product name, your batch number, your stage, sample stage, and who's sampled, and what kind of tests are requested and to whom you are requesting. So this is a typical format what commonly is being followed, and it changes with respect to organizations and industries. So it's a specimen copy of what we have presented here. You can understand this, and this is something like as you guys when you enter into the industries as a formulation scientist need to be generated once you prepare your composition or prepare your formulation style. Coming to a prototype formulations development. In each stage, a tablets or parenterals or any kind of a product, drug product, a colored product. So what all we have to do that? We have to generate a certain kind of prototype formulation development based on the dosage form. If it is a tablets, there are various techniques: dry granulations, wet granulations, and direct compressions. Dry granulations, as you guys you know that, like you can prepare a dry granulations by using a roller compactor, where you can mix your powder and feed into a roller compactor and get the flakes. Those flakes shall be broken by using a size reduction apparatus or inbuilt a screen into your roller compactor. And those dried granules shall be mixed with the proper lubricants and glidants and shall be filled into your capsules or shall be compressed into a tablet compression machine. A wet granulation shall be prepared by using a rapid mixer granulator or a top spray granulation. In this case, all the powders will be kept into the granulator. Externally, a binding solutions or a fluid shall be added while the granulator runs with certain impeller speed and certain shaft. So these two will optimize while doing the process and while addition of your spraying liquid or solutions. Direct compressions, which does not need any kind of granulation techniques. You can select a powder mixture with a uniform particle size, and if you need a sifting process, you can include the sifting process. and mix the components with the suitable glidants and lubricants, and directly you can fill it into the capsules or the tablet compression mixture to make it into a tablets. Similarly, parenteral also involves a process, mixing, filtration, a lyophilization process, a filling process, stoppering, sterilization process, and scaling process to be considered, or the drug product sequence to be considered while performing these all studies. Because the mixing happens from development to a bigger scale, it varies with respect to the shaft and with respect to your container sizes. The filtration process also changes because in development you do a filtrations with the small sizes where 47 mm or 142 mm, these sizes are being used, and when you scale it up you have to study a filtration effect, and as well as the validation also has to be done. Lyophilization process, it is something like freeze dry the product or freeze drying the liquid into a solid powder by using certain techniques, which is nothing but it follows a triple point techniques, where your frozen liquid shall be operated beyond your triple point. You can convert the product into liquid into solid mass by applying a pressure, time, and the temperature. And filling is one of the major factor where the liquid solutions are filled into your container, and it has to be very critical factor because you have to do a filling along with or without inert gas, because there certain products are very sensitive to an oxygen, where it is mandatorily has to be filled with a nitrogen along with the liquid, and followed by stoppering and sterilization. Sterilization is also is very important process for parenterals because it gives a product sterile in conditions to be used safely by the patients. Next, coming to certain critical products, like there are certain colored products also being used in parenterals. So those products also should be studied very well during the manufacturing optimization, because those colored product would stain on the most of the manufacturing vessels and in the manufacturing area, which basically we needs a dedicated facility, or you need a kind of once used techniques. Maybe you are from the manufacturing to until your filling line, you can use a plastic bags for the manufacturing, compounding, filtrations and filling systems. Maybe your needles and everything shall be reused for a specific product as and when it is necessary. Of course, when you handle this product, it is highly recommended to study these compatibilities with the drug products along with your vessels. We have discussed a selection or qualification excipients in our previous models. Hope I don't have to discuss in detail. The excipients always should be pharmaceutically acceptable and compatible, and as well as it should be regulatory authority should be there, because we have already indicated that it should be approved by the GRAS or the inactive ingredients database. An alternate dates also should be studied for our drug product along with the drug product, and the necessary stability has to be generated before we finalize our prototype formulations. Determination of process parameters. We have discussed few parameters in the previous slides. What are the parameters should be considered for the granulations, fluid bed processor, and rapid mixer granulators? Basically, the inlet temperatures, blower speed, a spray rate should be considered. A solution for injections, mixing speed, lyophilization process parameters like freezing rate, drying rate, vacuum, etc. should be considered. Here are just some examples we have presented It is case by case, a process or dosage form with respect to dosage form, these parameters varies accordingly. Let us look at how this inactive ingredients database, it is there in the public domain. You can get it with respect to your dosage form. For example, we have presented here acetic acid. Acetic acid also is available in different forms. Anhydrous citric acid monohydrate, and also it is being used in various dosage form. Buccal, intramuscular, intracardiac, and certain kind of tablets form also. And it has been defined how much maximum potency should be used in a unit dose and how much should be used per day has been tabulated. With this help, you can finalize your quantity and the route of administration shall be identified for the excipients. Look at the pictures of a coating machine where you can load the uncoated tablets here and do the spraying with the binding or the coating fluid. This is a rapid mixer granulator. You can add a rapid mixer granulator. You can add the powder. You can add the powder into the vessel and add the fluid on top of this with a controlled spraying rate. And this is something like fluid mixer granulator where you can perform the coating as well as top spray granulations. You can load the pellets on the bottom and you can do the bottom spray coating, and you can load the powder, and you can also use a top spray granulation technique also. Once you perform formulations or prototype development, you have to study the robustness of the formulations. How it has to be done, the robustness. It has to be kept in a certain accelerated conditions, storage conditions. If the product is stable at 2 to 8, then you can study the product at 2560 for certain predetermined intervals which has been defined in our ICH guidances. And if the product is stable at 25 degrees Celsius, you can study at 4075 as an accelerated along with an intermediate conditions. So these all you can very well refer it into the specific ICH guidances. Basically, we used to generate a 3 stability batches using 2 different lots of an API because the active pharmaceutical ingredients shows certain variabilities. Hence, always it recommends study 2 different manufacturer drug substances, and each manufacturer use 2 different grades of API. At least generate a 3 consecutive stability data. That will give you a full confidence when you transfer the product into your manufacturing site. Samples also should be predetermined time point shall be withdrawn and studied based on the dosage form test which we would be giving in our future models. Here are some more time points where you have to study for the long term up to 12 months. Minimum for summation data you need to cover it, and accelerated data at least 6 months, and intermediate for the 6 months. So these are the minimum study you have to perform when you submit a product to the regulatory authorities. Drug substances intended for storage in a refrigerator and freezer should be studied at least 12 months under refrigerated conditions and 6 months at 25-60 day conditions. And there are certain drug substances should be stored under minus 20 that has to be studied for 12 months as long-term conditions. So with this, we would like to close this model What are the importance and manufacturing conditions and optimizations are needed, we have discussed in this chapter. And also the prototype development for the dosage forms, followed by the stability storage conditions. We have developed and discussed shortly. If you wish to go through in detail, kindly refer ICH guidances, which we will be listing out in our reference page with respect to the chapter and the guidances numbers also. Thank you all. Wish you good luck.